Good afternoon. Welcome to seminar. Really good to see so many students here. This seminar features the last of our invited speakers, and it harkens back to the very first seminar last fall in terms of the topic area, which is molecular biology of plants. Dr. Ralph Dewey is an applied plant molecular biologist who got his PhD at NC State and then did a couple of postdoctoral stints at Rutgers University and the Waxman Institute of Microbiology. And he is now Philip Morris Professor of Crop and Soil Science at NCSU. He has a very diverse research portfolio. Early on, he was working on the molecular biology of soybean oil and use of cytochrome P450 genes to confer herbicide resistance. More recently, he has been working on drought resistance in ryegrass and a couple of topics that are more relevant to his seminar today: reduction of carcinogenic compounds in tobacco and modifying the tobacco genome through gene editing. So let's please give Dr. Ralph Dewey a warm welcome.
Hey, Dave. Yes, well, thank you for the introduction, and I definitely appreciate the opportunity to come and here at Kentucky and present some of the work we're doing in the area of harm reduction. I've had some very useful, interesting conversations this morning, and I actually feel that I'm giving my seminar for the second or third time, even as through the various topics I'll be talking about today, or we discussed earlier as well. And one of the reasons I'm excited to be here at Kentucky, as you'll see from my presentation, NC State and University of Kentucky have had quite a long-standing relationship and collaborative relationship in the area of harm reduction in tobacco. And a lot of some of the colleagues in the audience today had participated in one way or the other in some of the projects I'll be presenting. So one thing that I like, it's always easy about working on a topic like this, is I don't have to spend a whole lot of time trying to justify the problem. The fact is that even now, these data numbers may be a little outdated, but they'd still be pretty close to the truth. Fact is, worldwide, there's still around over 1 billion people or so consume tobacco products, mainly in the form of cigarettes, but then also in these other forms: cigars, BDs, pipe tobacco, smokeless products. Smoking attributes to over 1 million cancer deaths per year, including in some countries, about 30% of all cancer deaths can be attributed to tobacco use. So the importance of the area, again, don't have to spend a lot of time discussing. The solutions, however, become a little more complicated. Obviously, the best solution will always be, if you never smoked, don't start. If you do smoke, quit. However, the world that we live in isn't that simple, and we all know we're not going to wake up tomorrow and find that everyone's decided they're going to quit smoking. So there's definitely then a need, it's very important then, to focus on type of research strategies and development of products in which the toxic compounds that have been discovered or known, that the efforts can be done to reduce the exposure levels as much as possible. Again, I want to just totally emphasize, nothing we talk about today, no strategy could ever come out will be as effective as avoidance or cessation, yet the types of items I'll be covering can definitely help reduce on a population level in particular, reduce the tobacco-related harm for those who either can't or choose not to quit. So if you look in the area, it can seem daunting at first if you say, well, what is it in a cigarette smoke that is bad for you? The lists are pretty overwhelming, so we're not even going to try to focus in on this. But over 65 compounds have been described that are found in cigarette smoke that are carcinogenic or likely to be carcinogenic. So, like, where does one begin when the list is so vast? Well, again, there's been decades of research done on this, and I mean, it's definitely understanding, even though all these compounds here are found in cigarette smoke, safe to say not all of them are in fact contributing to cancers. I mean, you'll find some compounds here you'll find in apples, for example. It's all a matter of the concentration plus the potency. And there tends to be an inverse relationship between how strong a carcinogen it is and its abundance, the levels that you'll find it. In general, those compounds that are classified as very strong carcinogens tend to be found in very minor amounts. Those that are considered very weak carcinogens tend to be found in much more higher amounts. So it becomes very complicated to actually parse out or decide what specific compounds or combination of compounds are really leading to tobacco-associated cancers. However, through a series of, say, like meta-analysis type of studies, researchers have been able to get a better or more clear picture on which are some of the most likely participants, which are the worst of the compounds on the list. This is a study published back in 2003, still relevant today, in which, again, you do this sort of meta-study analysis on all the dozens and dozens of studies that have been done, whether they're animal studies, epidemiological studies, to say which of the compounds found in cigarette smoke are most likely contributing towards cancer formation. And this can vary according to cancer type. For lung cancer, for example, at the top, we have compounds, the compounds here are the ones considered the most likely to be involved within cancers of the lung. Here, cancers of the larynx, the nasal and oral cavities, the esophagus, liver, pancreas, and cervix. And what they've done is list the compounds here that are, again, the whole, or when you look at all the scientific data, are the most likely to be the worst players. One thing you'll notice on this list is the high prevalence of compounds referred to as NNK and NNN. You'll find like NNK, which is considered even a major likely contributor toward lung cancer, NNN, NNK to nasal, oral, and nasal cavity cancers, even those within smokeless tobacco product users, and the list goes on. So what are NNN and NNK? They are compounds that are within a class of a family called tobacco-specific nitrosamines. Now, and as the name would suggest, these are nitrosated products of compounds that are specifically found in tobacco. So everyone is aware, whether you've worked in tobacco or not, you're pretty much aware that nicotine is the predominant alkaloid found in tobacco. It constitutes about 90% of the alkaloid pool. The other three alkaloids found in fairly much abundance make up the rest of this 10%, nornicotine, anandamine, and anabazine. Now, each of these compounds, particularly most notably during the curing process, you find very little levels of these TSNAs in a mature leaf at harvest. However, during the curing process, it can also happen during storage, and even in some circumstances during the smoking combustion process itself. These tobacco alkaloids can be converted to specific nitrosated product. In case of nicotine, is ultimately can be produced to NNK, nornicotine produces NNN, anandamine, NAT, anabazine, or NAB. And again, dozens of studies have been conducted in animal systems and again, epidemiological studies as well. These have all shown that NAT and NAB are not carcinogenic. So no one's particularly worried about those. The same type of studies have identified NNK and NNN as being highly carcinogenic. And so these, when it comes to TSNAs and problems in tobacco, they're really talking about NNN and NNK. So the World Health Organization also takes sort of a similar view, trying to wade through the complexity of everything that's possibly harmful in a tobacco, and using and sort of summarizing the science in a way to try to identify what are the worst toxicants. And in 2008, they came up with this list. Again, we can't really read it here, but it doesn't matter, in which they were recommending at this time, they identified 9 toxicants that they viewed that the scientific evidence would suggest are being the worst players, and they were recommending that the regulations be written such to mandatory lowering of these 9 compounds. Again, kind of hard to see here, but the top 2 are NNN and NNK. Again, once again, they top the list. So in the US has no obligation to follow any recommendations of the World Health Organizations. In contrast, it's the FDA that has the regulatory authority over tobacco products here. And this began under the Obama administration in 2009. That's when Congress granted the FDA the authority to regulate tobacco products. This was called the Family Smoking Prevention and Tobacco Control Act. And included amongst the mandates was the ability of the FDA to regulate what would be acceptable levels of known toxicants. Along with known toxicants, they were also given the authority to regulate what the acceptable levels of nicotine would be. And I'll get into this a little later in my talk as well. Now, anyone who's had any association or familiarity with the FDA, particularly in the areas of tobacco, will realize that they move at a deathly slow pace. In 2009, when FDA was originally given authority to regulate tobacco, there was a lot of uncertainty in the tobacco industry. What's this going to lead to? What changes are going to be, the result because of this? And other than some early low-hanging fruit, like say regulations on flavorings that might be attractive to kids, or new, even tighter regulations on advertising, it's really kind of hard to say what the FDA has actually done or has accomplished in now 9 years. And they literally receive hundreds of millions of dollars for this program. And to say, well, what exactly have you done? A good example, one of the original things in 2009, I remember that's going to be one of the first on their list to discuss is menthol. Is it acceptable to put menthol in cigarettes? Here it is 9 years later, they still haven't made a decision. I think this has caused the tobacco industry to sort of maybe get into maybe a false sense of security that don't worry, nothing's ever going to happen, but this is a slow pace they're working. But there have been some recent events that suggest that maybe the FDA is actually going to get around to some of the mandates that they were given authority in 2009 to regulate as far as tobacco products. This came just a little over a year ago. It was published in 2017, where they took many in the industry by surprise when the FDA announced that it was going to propose to regulate the levels of NNN, that TSNA, one of those that we've been talking about, the levels of NNN in smokeless tobacco products. So for the first time, it looks like they're going to try to take some action on the idea of leveling mandating what the acceptable levels of a particular toxicant would be. And even though it's a huge, massive document, some of it can be distilled on this one sentence that comes pretty early, maybe in the first page. Basically says the proposed rule would require the mean level of NNN in any batch of finished smokeless tobacco products not exceed 1 microgram per gram of tobacco on a dry weight basis at any time through the product's labeled expiration date. That's kind of what this whole thing is all about. And again, this really got the industry's attention that the day maybe actually come when FDA will start mandating constituent levels such as this. And it looks like they're going to start with regulating NNN in smokeless products. So that's sort of a history on the history of sort of, well, TSNAs are association with cancers and what's the current regulatory status of the situation. Now we're going to go back in time, back to about 2001, where my lab began working in the area of harm reduction and tobacco. And our original, when we started working in this area, our original target was in fact NNN. And our strategy from a molecular biology viewpoint in conceptually is really pretty simple. And it's simply try to minimize the Nornicotine precursor. Okay, so here, if you look, nicotine, here's the structure of nicotine. Nornicotine is simply a demethylated nicotine, where this methyl group is cleaved off through the enzymatic activity of an enzyme called nicotine demethylase. And again, like I mentioned before, during the curing process, a portion of the nornicotine can be nitrosated to produce N-nitrosonornicotine, or NNN, the compound shown here. Again, I want to point out this is particularly a problem in burley tobaccos, the air-cured burley tobaccos. It's much more problematic, and the TSNA formation happens a lot greater under these air-curing conditions than you find in flue-cured tobacco. So most of what I'll be talking about is with the burley tobaccos, which are the type that are most of the work and breeding in Kentucky are involved with burleys. So the strategy was pretty simple. At this time, it was not known what's the gene encoding this nicotine demethylase enzyme. So our goal was to identify this gene, use molecular techniques to shut it down. We figured if we could shut down this gene, in theory, we should be able to lower the nornicotine amount, which then would, by definition, because it's the precursor, lower the levels of N and N. So this project I put into 4 different phases, and each of these phases spans multiple years. I'm going to cover some 15, 16 years worth of study here in 4 slides. Phase 1 was simply the isolation of the nicotine demethylase gene, a gene that is called CYP82E4. I'll just refer to this as E4 from here on. And this is, we define this as the major nicotine demethylase gene found in tobacco. We isolated this gene using a gene expression profiling strategy. The technology of the time, in this case, was microarrays. If we were to do this over again now, we'd use, like, say, an RNA-seq approach, but then microarrays were the most effective approach for this kind of gene expression profiling. Found it encodes an enzyme of the cytochrome P450 superfamily of monoxygenases, and also it's a member of a small, closely related gene family. We call E4 the major gene, but there are 2 other genes, one we call E5, the other E10, that we call minor nicotine demethylase genes that all encode this enzymes that can convert nicotine to nornicotine. And this is also from the very beginning our association with the University of Kentucky, as this project was done in collaboration with Balazs Saminsky, who is an assistant professor at the University of Kentucky at that time. Balazs, he was actually a PhD student of mine in North Carolina and a postdoc who then went from there to get an assistant professorship job at Kentucky. And at the same time this opportunity came about, we thought this would be a good way to continue collaborating on this project. Now we have phase 2, and that's testing our hypothesis. In fact, if we do shut down nicotine demethylase gene activity, can we effectively lower nornicotine and N and N? And to do this, we used the transgenic approach. We developed RNAi constructs directed against the 82E family. Nice thing about RNAi, even though you make the construct against one gene, closely related genes, it'll also chew them up and degrade them as well. And so we can sort of knock out the whole gene family. We found this to be, certainly met our expectations, if not exceeded them. We found that by shutting down this particular gene family in our RNA plants, we could reduce the amount of nornicotine that accumulates by about 80%, and the amount of N and N that is found in the cured leaf by a similar amount. This work was also done in collaboration with our colleagues here at Kentucky. We see here, along with Balazs, Lowell Bush, and Ann Jack, they were responsible for, say, conducting the field trials in Kentucky. We also had trials in Virginia and North Carolina. And also through a common theme for most of our projects is we have a great partnership with the analytical lab here. They do great work as far as analyzing some of these difficult to isolate and quantitate compounds that are involved. So step 2 was successful. Phase 3 is then developing a non-GM strategy to get the same effect. Now, a lot of people might be puzzled by the idea that the tobacco industry is concerned about GMs, but they are, just like any other industry. And in any case where they would be willing to consider using transgenics for a harm reduction strategy if that was the only option. But in a case like this, when you simply need to eliminate a gene function, there are ways to do this non-transgenically. And so phase 3 was, we generated these very large EMS mutagenized populations, a chemical mutagen EMS, large mutagenesis populations. We then grew up thousands of plants from this population, screened them using high throughput PCR and DNA sequencing to identify knockout mutations in both the major E4 and the minor E5 and E10 nicotine demethylase genes, and then pyramided these 3 within the same line. And we were happy to find that when we did combine the 3 knockout mutations in these nicotine demethylase genes, we are able to reduce the levels of nornicotine and NNN to levels just as good as our most effective RNAi transgenic line. So this was a good example of a case where we could use, say, transgenic approaches for proof of concept, identify the gene targets, and then move to a non-GM approach to get the same phenotype. So fourth phase is the variety development phase, in which we introduce, because like any commodity, you can't just say improve a trait in corn, make one variety, and then okay, you're done. No, you have to, if you want it the widespread, you have to put it into every popular variety that's used like throughout the country and even throughout the world. So in partnering with Ramsey Lewis, the excellent tobacco breeder at NC State, we worked on developing varieties that had these triple mutant, this low NNN trait, which our company partners, which was Altria and PMI, they gave this, they trademarked this name is the name Zyvert. We've never liked this name, but our industry partners seem to enjoy it. That's the name given this low NN trait. And again, we back-crossed this into 26 varieties here, including dark, burley, dark, and flue-cured varieties. And this is just what was done at NC State. Independently, Bob Miller here at Kentucky has developed Zybert versions of the popular burley varieties that come out of University of Kentucky, which do command the greatest amount of acreage of burleys. Also, some of our colleagues at Altria have also produced their own dark varieties for applications into smokeless products. For those who aren't familiar with tobacco, burley and flue-cured tobaccos are used to produce cigarettes. Dark tobaccos are used to produce smokeless products. Also, we took advantage of another connection here with Kentucky is molecular markers were developed by Dan Lee to accelerate this back-crossing project. And again, these varieties were all tested in the field. And the bottom line, this Zybert technology represents a very effective means of greatly reducing the levels of NNN in any type of air-cured tobacco, which means the burleys and the darks. The flue-cureds, it really doesn't make that big a difference, even though we introduced the trait in because of the curing. But it's a very effective way to greatly reduce the amount of this well-characterized toxicant in burley and dark tobaccos. So for those who aren't familiar with this story are probably thinking, okay, we can declare victory. The industry's probably out happily using this. We have one less carcinogen to worry about. But the fact is, as I just talked about in some of my discussions earlier today, right now, all these are sitting on a shelf. They haven't been used anywhere. And that's because the strategy, in order to do this very large-scale back-crossing program, and again, these were done back-cross the minimum of 8 times to bring things back to type, particularly when you start with a mutagenesis library where you have, you're just flooded the genomes with mutations. You have to do a lot of back-crossing to bring something back to type. To accelerate this process, we used a technique that Ramsey Lewis had helped innovate called the FT technology, in which you have the FT gene from Arabidopsis is put in, and it makes the generation time very, very fast. And it basically cuts the development of a back-crossing program, the time in about half from what a conventional back-crossing program would do. Then in the end, you cross away the transgene and just restore everything back to type. So in this case, there is no transgene in just these mutations that were produced by chemical mutagenesis. Now, again, our sponsors were aware of this and they were supportive of this through this whole process. When push came to shove in the time of commercialization, though, the fact that it used to have a transgene in it suddenly became a problem. In the US, this isn't an issue. We already have statements from APHIS, for example, saying that using this technology is not considered a regulated article. You can treat like everything else. The European Union, however, they have not wanted to make a decision on this. They've been into a perpetual state of, when it comes to any type of new breeding technology, whether it's things by genome editing, using the FT for there's a broad number of categories of called new breeding technologies that somehow, that maybe there was some time a transgene involved. So even though the end product has no transgene, they don't know what to do with it or how to regulate it. And as a result, there's been hesitancy to deploy these varieties, which is sort of a little, I find sort of a little difficult or frustrating in that, how do you regulate something that cannot be measured? There is absolutely no test you could give any lab to give a tobacco plant that had been, because the gene was in the past, it used to be a gene in it. There isn't anymore. You can give these materials to no lab that could ever possibly tell. So how do you regulate something like this? Anyway, that's kind of an aside. We are hopeful that sometime in the future, hopefully near future though, that they'll get over this hurdle, and I think particularly that the first deployment will come in these dark tobaccos that are used for smokeless products, particularly since Altria is mainly a US company with US tobacco, and these issues are not a US problem. And so I think that actually will be deployed very quickly in the dark tobaccos. So that was our original harm reduction strategy as far as how to reduce TSNAs. So we go back to our diagram here. There's fundamentally two different ways that you could reduce TSNAs in tobacco plants through modifying the genetics. The one, as I mentioned, is to target the alkaloid precursor, just like we did with the Nornicotine story. Reduce the Nornicotine, you reduce the NNN. The other strategy is target the nitrosating agent. If one could find ways to lower the amounts of the nitrosating agent that is found during the curing process, that would be a way you could, in theory, lower the amounts of all TSNAs globally. So in the case of air-cured tobaccos, like the darks and the burleys, the predominant nitrosating agent in this case is nitrite. And it's not nitrate, though, that's found in the leaf of the tobacco plant, because typically nitrite's a very toxic compound, highly reactive. You won't find it in very high levels in any plant. Again, because it's then very rapidly converted to ammonia and shuttled down the nitrate assimilation pathway. What happens is these burley and dark tobaccos possess a high level, they have high levels of nitrate. And during the curing process, leaf surface microbes will then convert that nitrate to nitrite. So this is really the microbial activity during the, say, 8 to 10 week curing period gives rise to the nitrite. This nitrite reacts with the alkaloids to then produce the TSNAs. So here are the theories: if we could come up with strategies to reduce or shut down the nitrate accumulation, this should then lower the production of nitrite, gives the microbes less to feed on, which in theory should lead to a reduction in all classes of TSNAs. So we've actually done two strategies in which we try to target the nitrosating reagent to reduce TSNAs. First strategy involves the expression of a deregulated tobacco nitrate reductase enzyme that I'll talk about in a little more detail in the next slide, in which the idea, if we just have an enzyme that will continuously reduce the nitrate and keep its pools depleted sort of continuously. The second is through the downregulation of a putative nitrate transporter gene. So, first strategy is this deregulated nitrate reductase activity. Research done back in the mid-90s, actually by Steve Huber, who was at NC State at the time, a plant physiologist. He did this work in spinach and worked on the nitrate reductase gene, and they show in nature it's a very highly regulated enzyme. Plants don't normally keep nitrate reductase turned on all the time. It tends to be on in the light, off in the dark. And it's also a highly regulated, not just at the gene transcription level, at the enzyme level. And most of the regulation is done through a serine at position 523, and the phosphorylation, dephosphorylation of this serine is the major determinant on whether the enzyme's active or inactive. It's found that if you substitute that serine with an aspartic acid, you will have an enzyme that's on all the time. And in this case, that would again be the best candidate for producing plants that maintain low levels of accumulated nitrate. And again, this is work done, as it can show here, collaboration Anjak, Jim Crutchfield, and Huihua is here as well. So here, just look at some of the most important points of this study. By expressing this deregulated nitrate reductase in tobacco, and here when it says the cut filler, that's just the unburned tobacco that goes into the cigarettes. It had again, this exceeded my expectations in the degree with which it would lower the nitrate. We would find, and this is very typical, in this case, like we only accumulated 6% of the free nitrate that you find in a normal or control plant, with no impact on the nicotine. Very dramatic reduction in the nitrate.
Where did you express the nitrate reductase?
35 S per motor.
But where in the cell?
It's a cytosolic.
Have you tried?
No, we did not target the vacuole. That is implicated in our next strategy. In this case, yeah, we just let it be expressed where it normally is expressed in the cytosol. And so if we look at the TSNA content, both of the unburned tobacco and then from cigarettes made out of the tobacco in the smoke, which is ultimately what's the most important from a health viewpoint, is what's in the smoke is what counts for a cigarette. Again, we find, again, we have the 4 different TSNAs here. In each case, in the unburned tobacco, we see a very significant reduction in the amount of the TSNAs. And we're most encouraged about NNK because arguably it's even worse carcinogen than NNN because it's one of the major ones that have been associated with lung cancer. And so we were really trying to target this particular TSNA more than anything. And one thing you'll notice too, that in everything except NNK, if you look at the TSNAs in the smoke, the differential between the transgenic cigarettes, I guess, and the normal is even greater. We see greater reduction in the TSNA in the smoke than we see in the unburned. And the reason behind it is like mentioned before, you can get some TSNA formation produced by pyrosynthesis. But the conditions in which this happens, research has shown back in the 80s, early 90s, is the conditions where you see pyrosynthesis is conditions where there's a high level of nitrate around. If you have high nitrate, you will get pyrosynthesis. If you don't, you won't. And so by reducing the nitrate, we not only reduce the TSNAs that were formed in the sort of cigarette before it was burned, we reduced its ability to form TSNAs in the smoke. And so if you look, again, about a 64% reduction of NNK in both the cut filler and the smoke. If you look at total TSNAs, we see about in the unburned tobacco, about a 52% reduction cumulatively in total TSNAs, which is about a 76% reduction in the smoke. So again, we were very encouraged by these results. And now the alternative strategy we used to focusing on the nitrate kind of gets back to David's comment on the vacuole, because that is where the majority of the free nitrate is stored, is in the vacuole. In this case, the target is a putative nitrate transporter gene. Now, as a little background, there's a family of genes called the CLC family of proton-coupled antiporters. They're called CLC because they're originally believed to function exclusively as chloride transporters. Work in Arabidopsis showed, I mean, it's a fairly large family, that some of these homologs are not chloride transporters, but are in fact nitrate proton antiporters that function to transport and sequester the nitrate into the vacuole. The most well-characterized Arabidopsis CLC is the CLCA, which again, in Arabidopsis, transports the nitrate into the vacuole. So the theory here is, if one could identify then a tobacco gene responsible for transporting nitrate in the vacuole, inhibit its expression, then the levels of free nitrate should be predicted to be produced along with the TSNA formation. The rationale here being that, again, in a wild type plant, a lot of the nitrate is protected from its endogenous nitrate reductase because it's in the vacuole. So if we can stop, inhibit this process, then it's still hanging out there in the cytosol where it can be exposed to the nitrate reductase enzyme and reducing the overall accumulation. So in tobacco, there are 2 genes, CLC-NT2 genes, we call them, like a lot of tobacco genes, since it's an allotetraploid species. One of the genes originated from the sylvestris ancestor, the other from the tomatosiformis ancestor. 2 of them, they are 786 amino acids in size. They're 98% identical to each other and 77% identical to this Arabidopsis ATCLCA nitrate transporter. And so again, using sort of the same strategy with nicotine demethylase, the idea is use transgenics for proof of concept, and if it works, then you can go on to the non-transgenic strategies. So we developed an RNA construct to determine the effect of suppressing gene expression on the leaf nitrate and TSNA formation in burleys. And we have 2 years of field trials. I want to summarize some of the highlights. In the 2015, here we tested 5 different RNAi suppression lines compared to the wild type control. The 2 best lines ended up reducing nitrate to about 80% compared to the wild type control. not as dramatic as our overexpression. That went all the way down to 6%, but still, or I guess 94% reduction, but still very significant reduction in nitrate. And as far as with TSNAs, saw this year anywhere between a 40 to 50% reduction in NNN, NNK, and NAT in the cured leaf. Moving on to 2006, we repeated using the best line, we repeated these results. And so in this case, we have our wild type controlled, the Berberine variety Tennessee 90. This is the same variety with our best RNAi anti-CCL NT2 gene. And this year we saw 73% reduction in the nitrate and 44% reduction in NNN, 48% reduction in NAT, 21% reduction in the NNK. So again, another method that can be used to significantly reduce the amount of TSNAs. This sort of has an advantage of the first strategy that I described, in that one can theoretically make a completely non-GM version of this particular technology. So I just want to now summarize the 3 different strategies of TSNA reduction that we've been involved with and what's sort of the current status. So the first one being this, for our nicotine demethylase gene activation, the target here was solely NNN. Here I have, say, timeline for development of efficacious varieties. In this case, it's already done. I mentioned it's kind of just sitting there. And these include 6 dark varieties that could be suitable for production of smokeless products, which I think will be the first that are commercialized. The other 2 strategies, the deregulated nitrate reductase expression, targets both NN and NNK. Here, we'd advance by the time it would take, say, to breed these in and do the field testing, variety development, a 4 to 6 year period. But as the asterisk would say, this strategy requires the plant either to be GM or non-transgenic using new breeding technologies like genome editing. The last is the nitrate transporter deactivation, which again targets NNN and NNK. Here, the timeline to develop varieties that could be good for commercialization. I say 3 years to 8 years, the 3 years being based on our ability to use new breeding technologies like FT to actually backcross and develop the new varieties. The 8 years would be more like what it would take if we have to, again, have our hands tied and not use modern technology to produce the tobacco variety. So with this, I want to move on sort of the next topic area, and that is nicotine reduction, which again is something that has gained a lot of popularity lately. For reasons even different from what I'm talking about today, I became interested in this area several years ago. I thought there'd be utility in developing high quality tobacco lines that produce very little nicotine. I remember even here in Lexington, there's what's called the Tobacco Workers Conference. It was held here a number of years ago. I had my student giving a poster of the progress we're making on developed low nicotine tobaccos. The response was, "Oh, this is interesting," but not a lot of people cared, frankly. Now it's the most highly talked about topic in the tobacco industry today. Let's first though back up and say, well, why would one want to even create a low nicotine tobacco? Isn't that why people smoke? Or to use tobacco products is to access the nicotine. So here's the potential applications. One is actually use a smoking cessation tool, and there's a small company called 22nd Century that we work with them. This actually for years has been their business model. And I was kind of skeptical at first, but actually a number of studies have shown, and a lot of these have been sponsored by like the FDA or NIH, that have shown that people who go a period of time, people who want to quit and go for a month or two smoking a very low nicotine cigarette, it helps them wean them off the habit and makes it easier to quit. And these are studies published like in the New England Journal of Medicine and all, and it is repeated then several times. And so there actually might be something to this idea. Another alternative use is the platform for alternative uses of tobacco. There's over the years a number of groups, or there's been interest in this idea of production of pharmaceuticals or vaccines in tobacco, or even using tobacco as a biofuels feedstock. There's even a startup company called Titan that this is their whole goal. In both cases, it's much more advantageous for them to do this in a nicotine-free background, whether it's saving costs in purification, or in the case of biofuels, the nicotine inhibits the yeast required for fermentation. So these are some other applications. The one getting the most attention is this bottom one, the possibility that the FDA or the World Health Organization may mandate future tobacco products have lower levels of nicotine. The kind of talk in this area began to first be ramped up in 2015. And this idea had been thrown around for decades, but no one ever, I think, took it very seriously. Then in 2015, the World Health Organization, they published this work group study in which their recommendation came out, very strong, that they recommended that nicotine levels should be mandated to be lower, below a level that would give an addictive response. And that's really quite low. It's got to be about 5% of what it currently is to get it low enough where it won't initiate or maintain an addiction response. That was in 2015. Just last year, on January 28th, the FDA kind of shook up the tobacco industry by their own announcement that the FDA proposing plans in which they are, again, like the World Health Organization, proposing the idea of mandating dramatically lower levels of nicotine in cigarettes. Again, the idea is sort of written in small type. The idea is to have nicotine levels in cigarettes be reduced to a non-addictive level, is the plan here. It still is a huge leap still to say whether this will, if and when this will ever be implemented, but it's definitely caught people's attention. And this is a big focus in the whole entire tobacco industry, is what to do if the FDA, if this truly becomes a future mandate. So how does one make a low nicotine tobacco? You can do it by conventional breeding. In fact, there's a couple of loci discovered in cigar type tobaccos, I believe in the 50s, they're called NIC1 and NIC2, that when combined, they can greatly reduce the amount of nicotine. The industry and researchers have played around for these for a few decades. It's effective in reducing nicotine, yet a large number of genes are altered. There are quite sizable chromosome deletion mutants affecting genes beyond just alkaloids. They affect yield. They don't ripen properly. And the bottom line is they're low yielding and they're poor quality. And they're not very usable for the industry. The other approach would then be molecular breeding, which would largely entail targeting mutations in specific genes of the nicotine biosynthetic pathway. So if you look at this is actually a simplified version of the nicotine biosynthetic pathway, but it's basically nicotine comes formed through the conversion of two separate metabolic pathways. One's called the pyrrolidine ring pathway to produce this pyrrolidine ring here. The other is the pyridine ring pathway, which then at the very end, the two rings are joined to form nicotine and related alkaloids. The steps here shown in gray are those that in the past have been isolated, characterized, and that researchers have used to try to downregulate in attempts to make low nicotine tobaccos. There's problems every time you try to do this. Anytime you inhibit one of these genes of the pyrrolidine ring pathway, what happens is the other pathway is still active. The metabolic flux all flows down, and you get a big increase in this normally obscure or low-level alkaloid, anandamide. So yes, you have low nicotine tobaccos, but now you have tobaccos that produce a large amount of anandamide, of still fairly largely uncharacterized alkaloid. So no one wants to go the direction of trying to propose producing tobacco products with some obscure, not well-characterized alkaloid. Problem with up here, this QPT step, it's a gene that's not only involved in nicotine biosynthesis, but it's part of primary metabolism as well. It feeds into this pyridine nucleotide cycle, produces the essential cofactor NAD. You can only shut this down so much to reduce nicotine before you're hurting normal plant growth and development, because you're restricting its ability to produce NAD. So the gene that we thought was the most promising for producing low alkaloid, high quality tobaccos was the latest that was characterized and discovered, one referred to as BBL. which is arguably is probably the last stage of the nicotine biosynthetic pathway. It encodes an enzyme that cannot be known successfully been able to conduct or devise an in vitro strategy to definitively define what the reaction is, but it's very clear it's at near the end, if not the very last step of nicotine biosynthesis. This gene was first characterized in 2000, and I think 11 is when this publication came out by the lab of Takashi Hashimoto in Japan, where the BBL stands for Berberine Bridge Enzyme-Like gene family. Hashimoto's lab characterized 4 different members of the BBL family, called BBL A, B, C, and there's a D, but he reported very minimal expression. That the major ones are A, B, and C. They encode a flavin-containing oxidase, but again, the enzymatic function is unclear because no one can define an assay to do this in vitro, but it clearly occurs at the very end of the pathway. So we then followed sort of this similar strategy that we talked about before. We used transgenics to shut down the family to see if it could, the degree with which it could lower nicotine, and it did. It lowered it very well. Then we went with and screened our EMS mutagenesis library, pyramided the mutations in the 3 major BBL genes, A, B, and C, into the same plant. And that's what we have here, is that when you pyramid, knock out mutations in the 3 major BBL genes, you can then lower nicotine levels by more than 90%. In this case, also done in burley tobacco background. Now, our current efforts in this area are to use this is getting close to where we want. Like I said, I think ultimately we need to reduce it about 95% to meet this threshold where it would be compliant with FDA's proposed standard. So we're using now genome editing strategies to reduce the levels even further. Again, ultimately we'll be able to produce like high quality and low nicotine tobaccos that could be suitable in case this ever comes about with the mandate by FDA or World Health Organization. Final topic I want to then just touch a little on is the efforts we're doing in the area of genome editing. As many of you might be aware, it's one of the sort of the hot areas these days, and probably the most innovative or transformative technology in molecular biology since PCR, in which it can enable precise subtle genome modifications in a manner that ultimately leaves no foreign DNA in the end product. So there's both technical and what you call regulatory or societal advantages to genome editing over traditional transgenics. From a technical viewpoint, in many cases, there can be a desire, the phenotype you want in a crop species, whether it be tobacco, corn, or whatever, can be achieved by, it doesn't require the introduction of a whole foreign gene. It's just more of a subtle modification of an endogenous gene might be all that you need. And also, one downside of these traditional mutagenic agents like EMS, which we've used, is again, they cause genome-wide perturbations. They're not targeted, so it really messes up the whole genome, and it takes excessive back crossing to get rid of them. And also, many desired mutations are simply too complex to recover using these non-selective agents. If you just want to knock out a gene or make it non-functional, okay, you could do it with the traditional mutagens. What if you want to change a single amino acid on a protein? You'll likely never achieve that. But this is what prompts the ability of genome editing technologies. From a regulatory manner, is if plants generated in this manner are not considered regulated articles, and APHIS has delivered a number of statements saying for the most part they don't consider these regulated articles, then there's huge costs and time savings associated with commercialization compared to traditional transgenic crops. And there's, at least within some sectors, the possibility of getting more public acceptance since the end product contains no foreign DNA. The key to precision genome editing, it relies on the introduction of a double-stranded break in the genome at a precise location, and the fact that double-stranded breaks are repaired within the cell by one of 2 distinct mechanisms, either non-homologous end joining, which can be sort of viewed as the cell trying to religate, stick the pieces back together, The other is through homologous recombination, which is the repair is corrected by having a homologous template in an endogenous situation be like a sister chromatid. In an engineering one, it's sort of a donor template, a correcting template that you can, the researcher introduces. In this case, the repair is very accurate. The problem is in your typical plant cell, the non-homologous end joining machinery is far more prevalent and easier to tap into than homologous recombination. So there's three potential applications for using these designer nucleases. One is to achieve and create simple gene knockouts. And this is where you put your designer nuclease in, and then this imperfect repair by non-homologous end joining will lead to small insertions or deletions that knock gene function out. This is really quite efficient. The other is what's called gene knock-in, is where you could actually introduce a foreign gene or make a transgenic plant where you put the transgene in a location that you want rather than just having going at random, like is historically how it's done. We're not particularly interested in this, but it's more the end to. The last being this gene surgery, which is, in this case, it still needs a homologous recombination, but in this case, you have a correcting template that's just introduces subtle nucleotide substitutions in to create, again, much more subtle differences like changing an amino acid in an enzyme, changing an element in a promoter sequence, and again, which we call gene surgery. So there are actually 4 classes of editing enzymes that have been characterized. A lot of the attention is all focused on this CRISPR-Cas system. That's what's gotten most of the publicity lately. But there are times and places I'll describe where some of these other enzymes should be considered. These include custom designed meganucleases, zinc finger nucleases, and talons. So in my lab, we're using genome editing for the purposes are, again, to introduce some of these harm reduction traits to lower either carcinogen levels or nicotine levels in a way that there's no transgene left in the end. One project can inhibit lateral meristem formation, which in the tobacco world means help inhibit suckers that formed after the plant is topped. And also you can introduce herbicide tolerance trait. And for example, you can generate a Roundup Ready or a glyphosate tolerant plant by merely introducing 2 amino acid substitutions in the endogenous EPSBS synthase genes, and so not require, say, a whole foreign transgene coming in. The genome editing systems we use are CRISPR-Cas9. This is our system of choice for basic scientific studies or when we're doing proof of concept experiments. We also use custom designed meganucleases through a collaboration we have through Precision Biosciences, a company that's in the RTP in North Carolina that develops these type of enzymes. And these are the systems that we use when our sponsor requires freedom to operate in the end. And our initial problems with the CRISPR-Cas9 was, and it's still an existing problem, is who owns the technology. You have University of Berkeley suing MIT, the Broad Institute, and you have even a number of other players. Even though patents have been issued, it's not really settled until they've been vetted in the courts. And it's been predicted it's going to be years before the ownership is ultimately decided and who do you even go to get a legitimate license. This is the region I was sort of steering some of our corporate sponsors towards Precision Biosciences because they don't have any of these issues. They have a strong patent portfolio. It's been vetted in the courts already. And so there's no question if you do genome editing with this technology, you're not going to have freedom to operate issues. That was the original issue. And an additional one came up recently that again, of why we would stay away from CRISPR-Cas9 when you want to use the end product was a statement that was put out in September 2016 by the Broad Institute at MIT. And this is the group that has arguably the strongest IP portfolio in the whole CRISPR-Cas9 system, where they declared that they would not grant licenses for the CRISPR technology to be used in tobacco. There's 3 specific restrictions they said they won't license their technology. One is to create a gene drive, the other sterile seeds, in other words, to replicate this terminator technology, or used in tobacco, unless you're just doing model organism for research, or non-tobacco, like if you're doing pharmaceuticals, for example. Otherwise, they will not license it to tobacco companies. And this then became another reason that we needed to say alternatives to CRISPR-Cas9 if we're working in tobacco and we want to actually produce a commercialized variety one day. So our editing results so far, as far as using either CRISPR-Cas9 or these meganucleases, they both work really well in our hands for targeted mutagenesis. To date, we've mutated a dozen different tobacco genes using CRISPR-Cas9, 7 using these custom designed meganucleases. And again, both work equally well for targeted mutagenesis. What's more difficult is the gene surgery. And this is the type of thing that we want to do to, like say, one of our main goals is to introduce this in our nitrate reductase gene, this amino acid substitution of position 532 to create sort of a non-GM, a genome edited version of our low nitrate, low TSNA plants. So to summarize and finish up here, our current genome activities focus on this is we are working on developing procedures to get efficient homologous recombination mediated gene surgery. And we're convinced this is best done through adding the editing constructs and correcting templates into protoplasts and regenerating whole plants out of protoplasts. And this is not the easiest thing to do, but we've actually happily been successful in developing a pipeline of protoplast regeneration tobacco that's actually working very well. The other thing we're doing is working with Rodolphe Barangau, who is one of the innovators of genome editing technology. Work he did at DuPont was some of the seminal work in describing how CRISPR-Cas works within the bacteria. The information was then used by those who then co-opted it as a genome editing tool. He is now at NC State, and he's working with our tech transfer office to develop novel or unique CRISPR editing tools that'll be controlled by, the IP controlled by NC State, not the Broad Institute at MIT. Specifically, we're repurposing CRISPR components from a lactobacillus, one called Lactobacillus gasseri, to function as genome editing tools in plants. And the goal here is to hopefully give another option for, say, a CRISPR type of technology that could potentially be used in tobacco, given how the IP holders of the main technology won't let it be used for tobacco. Finally, just want to acknowledge the members of my lab working on the current members are working on these various projects I've talked about. The lab of Ramsey Lewis, that everything is in very tight collaboration with our tobacco breeder Ramsey. Rodolphe again for developing new genome editing systems. We have a very active collaboration with Lucien Beauvais and Simon Gofert at PMI. And of course, obviously want to acknowledge our ongoing collaboration with the researchers here at University of Kentucky. And with that, thank you for your attention.
Questions for Ralph?
Anne.
The 22nd century of the cigarette. Do you notice that is a blended cigarette or filtered?
It's my understanding, and again, I don't know if this is 100% true, because when it comes to industry, you don't know whether you're fully getting all the right information. My understanding is they are taking a burley and flue curing it. and you can probably guess how good they taste when you do that.
I'm just wondering if the reason that people appear to quit is not because they're different addictions.
We've had that same conversation, and I've had that, just earlier today, and I've had that same thought. Again, we talked to them quite a bit, and because you're right, I think most people agree their cigarettes taste pretty bad. So once the experiment, I guess, ultimately will be found, because what we're developing together with them should be fairly normal-looking tobaccos, like a flue-cured low, not having to rely just on pure burley. So it's safe to say that ours, the ones made from the tobaccos we're developing, will likely taste more like a normal cigarette. The degree with which that influences then people who want to quit, I guess we'll find out. I mean, that idea has definitely crossed my mind as well.
Yeah, I think it's pretty significant because the FDA is basing the level of nicotine that they're studying to regulate on these.
Of course, the FDA motives are different than 22nd Century's original one. They set a model to try to help develop a smoking cessation device. FDA just wants to get it below an addictive level. Okay, okay, we can. It's likely in theory we could do that. So it might be we could what we're developing together with them. Maybe it won't be that great for a smoking cessation tool, but it might work perfectly fine if the goal is to meet FDA compliance. Which still, they kind of go hand in hand, because if you get it below an addictive level, then, well, it should at least meet the goal of they're not staying addicted to them because of the nicotine, because it's below the level that can maintain it.
But like Richard, is that a realistic system of that list based on these cigarettes?
The level, I mean, there, again, I still think we have two different issues. Can you achieve the level? And then what's the consequences of, and again, 22nd century is pushing from the idea that this level will help people quit. I don't think FDA is necessarily looking is that it will help people quit in as much as they'll never get hooked in the first place. If they, you know, if that's the only option you have, or it won't maintain, and so they'll naturally stop using it if it's not giving them the sort of nicotine hit. George.
Pleiotropic effects of the kind of knockdowns of the nitrate transporter.
Good question. I think it'd be a natural thing. Well, if you're just forcing a plant not to store much nitrate, isn't there a consequence to this? It's interesting. There are some of which, again, everything we do basically is sponsored by industry. So some things, everything obviously I spoke about today, I can talk about. Others I can't. But to answer your question to the level that I can is yes. There is definitely with the first technology, not much with the level. And I think it depends how much you're driving the nitrate down. We drive it down much further, lower levels with our first strategy than the second. I haven't seen much noticeable with the second strategy. The second strategy with the nitrate transporters, of course, doesn't quite lower the TSNAs as much either. The first strategy, yes, there's definitely a yield hit, but we've been able to overcome it. We can get around it, we can overcome it, and it's the how we overcome it is what I can't talk about. But we can end up creating normal looking tobacco plants with the low nitrate, with the deregulated level. We can, again, overcome that hurt, the initial negative effects that are caused by, again, forcing the plant to always funnel its nitrate downstream. David.
What effect do people see plasma season if that exists below the
Yeah, there's again, quite a lot of history about this, just even with study this NIC1 and NIC2. The bugs like it more. I mean, the ultimate consequence, the ultimate answer is, yeah, you have to spray it with more pesticides. Because, yeah, nicotine's obviously a very strong insecticide. And so if you don't want the bugs to eat it even more, you got to spray or maybe use some different pesticides that you wouldn't otherwise have to use when growing tobacco. Yes, Orlando.
The nicotine level is fairly consistent in the field from the transgenic.
Well, there's going to be year-to-year variability, but I'd say we don't have enough data yet. We don't have enough field. I mean, a lot of the work with low nicotine is still, you know, like when times generating pyrimidine mutations and all. This is a work in progress. We don't have enough in the field to say what the extent of variation is. There's clearly going to be some. It is, and right. Again, whether we can, I mean, again, this is a question that we will hopefully know within the next year as we continue this project. Can we create it to a level that's consistently under most environments and most years tends to be beneath that threshold? That's an open question. We can drive it down very low, but same issue with the NNN proposed by the FDA is putting out. Can you always maintain that level? I mean, that's trickier, but you know, we'll see. George again.
What's the on-target specification specificity mechanism involved in making
So what they did, the whole concept of a precision biosciences, they start with a naturally occurring restriction enzyme from Chlamydomonas. It functions in the chloroplast of Chlamydomonas. It is meganuclease is a sort of name. Homing enzyme is another name given to this class of enzymes that have naturally long restriction recognition sites. And so in this case, it's a 22 base pair recognition site by this ICRA1 is the name of that homing enzyme. So their whole company is built on protein engineering that you say that they've obviously done the 3D structure, they know the DNA contact points, how to modify those DNA contact points to recognize different bases. They had a decent but modest level of success when they're going purely by rational design in the re-engineering. What's taken up to a whole other level is when they changed the whole model to one of directed evolution. Now they can, when I first started working with them, the technology was not very mature, and we'd give them a list of 10 genes and they'd say, oh, we think we can make one against one of them. But since they gone to this directed evolution mechanism of re-engineering the enzyme, we can give them any gene we want. I don't think that's any more restrictive than any of these other genome technologies as far as giving a gene sequence, having it differentiate between two closely related isoforms, cut one and not the other if you want, or conversely cut both if you want. But yeah, ultimately it's the re-engineering of a naturally occurring restriction endonuclease. And I do that by pushing the through my mic. Okay, so Princeton, do you have any questions? I didn't press this. I was supposed to press this other button too, I thought.
I think that's probably okay.
No, we don't have any questions. I don't. Do you, Andy?
Okay, they heard.
No, I'm good. Thank you, Ralph.
Oh, sounds like Andy. You're welcome.
Yes, thank you.
Anything else from Ralph? I wonder, what changes occurred to NUCLEASES in
What changes occurred in this is a question that we wanted to study. What's the mechanism? I think you're asking what's the mechanism for what makes the E4 allele where in its normal state it is essentially inactive. In a converter state, it suddenly now is expressed at very high levels. And so obviously one speculates is epigenetic. Well, first of all, the first Earl Wernsman years ago, he speculated there's a transposable element in there. That would be a good theory. But once we actually identify the gene, sequence the genomic region, clearly there's no transposons anywhere in here. So then you figure, okay, and in fact, we did like broad DNA sequencing over the E4 in a converter, strong converter, non-converter, no difference in nucleotide sequence. So then you go to the epigenetic models. We did as much at the time of bisulfite sequencing to determine the methylation pattern, converted, non-converter, because again, the next obvious thing is, well, maybe the methylation state has changed. We couldn't find any difference in the methylation pattern, but it's not as exhaustive as you can do it today. So we covered major regions to determine what the methylation status was, but we're not ruling out that option still because we weren't able to comprehensively cover the full. Eventually we dropped it because, like I mentioned before, I mean, it's a very interesting scientific project. It's still kind of like to know the answer to that question, but industry doesn't care about answering questions like that. They give money to solve a problem, not to figure out a scientific phenomenon. So we stopped working on it after we did the methylation sequencing for.
how many people do you think